around 1000 structures. These 1000 structures need not to be synthesized in the physical world, okay, by paying a lot of money and also by spending a lot of energy from different people. It is not required. Simply do the molecular docking studies. Out of 1000 molecules, you select only 20 molecules. If you have enough facilities, select 50 molecules. Synthesize those 50 molecules only based on some rankings which were given by molecular docking technique, and based on that, these 50 molecules definitely will be successful molecules, because now you have some rationality for picking these 50 compounds. Now you have some support saying that yes, this compound will properly bind onto some target molecule by forming some interactions. Okay, so the interactions I just mentioned there, like shape complementarity and chemical complementarity, because you know there is a specificity and selectivity when it comes to some target molecules. Similarly, the same thing you can reproduce it to some drug and excipient also. For example, excipients you have polymers, you have various polymers. It could be PLGA, it could be simple like cyclodextrin. Okay. For example, you want to do cyclodextrin inclusion complexes. So how you will take decision? Like for example, there are close to 30 cyclodextrin materials are there. Cyclodextrin derivatives are there. Okay. And what kind of polarity you want actually to make your compound into a proper cyclodextrin inclusion complex. So for doing that, out of these 30 polymers, you can simply do a docking study, and whichever is giving the best score, there are different interaction energies are there. Van der Waals interaction energy, hydrogen bond and hydrophobic interactions are there. So based on that, now out of these 30 polymers, we will select one particular polymer, or maybe take two. Nothing wrong. Okay, for example, beta cyclodextrin very cheaper. Don't worry, you can get anywhere. Okay, but the other derivatives are very costly. Even 1 gram itself may be costing around close to 12,000 rupees. Affordable, still affordable. But I saw there is one cyclodextrin, I think so propyl, some side chain is there. Acyl cyclodextrin, they are cheaper only. But propyl, which is blocking the hydroxy groups, which is costing 1 gram itself 34,000 rupees, which is not affordable for M.Pharm guys or even for PhD students. Okay, even industry can afford that particular kind of cost. But when it comes to research purpose, it's very tedious process. So before doing any real-time activity like real-time interaction analysis, real-time inclusion complexes, before just do molecular docking and confidently select that 2-3 polymers, purchase them, and definitely you'll get close to 80% of success rate. I'm not telling 100% success rates you will get, but there are chances of getting close to 80% of success is there. Okay, you can select that 2-3 polymers and you play with them, do different kind of formulations with different ratios, and definitely you will get a proper formulation having that inclusion complexes very well, that you can formulate into next nanoparticles or liposomes, leosomes, whatever different formulations you can do. Okay, that kind of ratios we can take based on this protein ligand. Here, that ligand is drug molecule only or any chemical entity. The protein can be anything. The protein can be a polymer, small polymer like cyclodextrin, or even bigger polymer. Sometimes you can take dendrimers as a polymer. The dendrimers you can take, and drug loading you can check. How many molecules will be accommodated under that particular dendrimers? Is it first generation, second generation, or third generation dendrimer? How many side chains are there? So based on that, you can load the drug molecule. What is the efficiency? Now you can load the drug, whether the drug will be properly released or not. That also you need to take care of that. And these kind of things you can do in protein and like molecular docking wise. Just now I told you about that particular interaction. There are 4 types of different interactions are possible, starting from hydrogen bond, which is very important for pharmacology and chemistry, but may not be important for Sutix perspective. In Sutix, we will be looking for hydrophobic and electrostatic interactions, because we wanted to release a drug molecule from the polymer, but the polymer has to actually have some tendency to catch hold of it for certain extent of time, because we need a proper stable dosage form. If you are keeping in room temperature, after 3-4 days if it is coming out, then there is no use of the formulation. So we need to have proper interaction, proper energy between both the molecules. You should be having some stability over a period of time. Okay, so these type of interactions we will be seeing, and this is only molecular docking. And beyond that also we have something called as molecular dynamic simulation. So molecular docking, actually 3 types are there. Maybe I am taking 1 more minute to explain that. There are actually 3 concepts are there in the molecular docking. One is rigid docking, second one is flexible docking, the third one is semi-flexible docking. Okay, so rigid docking is nothing but whatever I told you. In mobile you can operate simply. The protein structure will be there. It is in one particular conformation. Okay, basically in biological system, proteins are always dancing. Protein is not a stable structure. You might have studied in biochemistry or somewhere in organic chemistry. Protein is a dynamic molecule. Either it is in the cytoplasm or it is in the blood. Okay, if it is in the serum or cytoplasm, doesn't matter. It will dance always. Some proteins are in soluble situation, but still they have a complex structure. Okay, they're always moving. Okay, for example, if you're doing a rigid docking where you're taking particular structure like this, it is fixed one now. You're not giving any flexibility to move this molecule into a different orientation. For examination, this is a drug molecule, so it is coming and binding, that's it. But this is a rigid molecule, which is not giving the actual bioactive conformation of the protein. So you have to give some flexibility. So giving flexibility and make sure that you have a good computational capacity. It won't run on mobile, forget about mobile. It won't even run on some computers i3 and for example you have Ryzen 3. If you want to run flexible docking, you need to have a gaming laptop. It won't happen in normal computer. Only flexible and semi-flexible docking can be done in normal computers. Sorry, rigid and semi-flexible can be done on normal computers. But if you want to run flexible docking purely, if you're giving entire freedom to the structure, it will take long.